In this video, we will discuss about how this receptor activation is also affected by the induced proximity of the cell surface receptors. We will start with this panel A. In this panel A, first on the left hand side, it shows how the natural cytokines interact with the cell surface receptors that will trigger this natural interleukin-2 signalings. And these things are usually occurs in the immune cells where three receptors comes together in order to trigger this natural interleukin-2 signaling, that is interleukin-2R beta, interleukin-2R alpha, and interleukin-2R gamma. In this interleukin-2R alpha usually occurs in a specific conditions that is related to the how much immune response is required, and that will cause the interleukin-2 signalings, and through the phosphorylations of this STAT5 JAK1 JAK3 complex that will cause the phosphorylations of the STAT5 to go to the downstream towards interleukin-2 signalings. In normal conditions, the proximities of the interleukin-2 alpha, 2R beta and 2R gamma is not enough in order to trigger these downstream signalings. So there are some synthetic bispecific antibodies are developed that can interact with this interleukin-2R beta and interleukin-2R alpha, that is cytokines mimetic. It is able to which is commonly called the cytokine mimetics, which is able to mimic the same effect of this natural cytokines, that is activating the downstream signalings, that is synthetic interleukin-2 signaling activations through this JAK1 STAT5 JAK3 complex formation and phosphorylations of this STAT5 to go to the downstream processing. So these different kind of cytokine mimetics are available that are shown in this panel B, where we can see that there is single chain FV and single chain FV79. This can also be used. The dual bispecific antibodies, and this is the unique antibody, uni antibodies, and this IAB80 and this contrast body antibodies. So, and this is not only limited to interleukin-2, but this bispecific antibodies are also formed with interleukin-2 and interleukin-10 antigens. And sometimes what people have find out is that not only does the interactions of the cytokines to the interleukin-2 is important, but in a right geometry the interactions need to be occurs, and that is shown in this panel C, where the no signaling occurs if the interaction is not occurs in the right geometry. We can see that the downstream intercellular components are not phosphorylated because the upstream interactions is not occurs in a specific manner, or not, in this case there are no antibodies right now present, even though they are in a close proximity, that is KLB and FGFR1C. But when these antibodies, that is anti-KLB or FGFR1C or FG21, which is the natural cytokines, when it interacts with this in this region, it will cause the downstream signal activations to the phosphorylations, and that will trigger this FGFRC1 formations, and that will cause this FRS2 PLC gamma STAT1 signaling activations. This is how sometimes geometry of the interactions is also important. Even though the receptors are in the close proximity, they won't trigger the downstream signaling because the geometrical interactions is not enough to cause the intercellular changes. Next one is about this WNT pathways. So since the receptors are far away from each other, it won't trigger the downstream signalings, and in this case the downstream signaling is not triggered. That will cause the continuous beta catenin deactivations or degradations, not deactivation, degradations through this complex formation of axin APC GSK3 beta catenin phosphorylations. But when these WNT surrogate antibodies are used, or WNT in a natural case, it will cause the FZD and LRP5 LRP6 to interact, and it will cause downstream signaling activations, that is it will prevent this complex, that is axin APC and GSK3 phosphorylated complex, to interact with the beta catenin. In this case beta catenin will not degrade, and it will goes to the nuclear localizations and trigger signalings. So this in the panel E, in place of this WNT surrogate, different kind of WNT surrogates are shown this panel that has been developed. That is SCV, SCFV, DKK1, diabodies, DAR pin, DKK1, SCFV, FCs. So these are some of the WNT surrogates that can be used for this intercellular processing, that is activating beta catenin or preventing beta catenin for degradations. And in the last panel is about this natural PTH signalings. So natural PTH signalings is usually occurs when this PTH is bind to this receptors, is PTH1 PTHR1 receptors, that will cause this complex formation in the downstream part. This G alpha S, G alpha Q, beta arrestin. It will cause the upregulations of the CAMP and Ca2 plus, that will trigger natural PTH signalings. But when these PTH receptors are far away from each other, the induced PTH receptors can be used. The PTH, we can say that synthetic PTH ligands can be used, such as PTH, VHH, that can helps in the interactions of these two receptors, and it will cause this G alpha S and G alpha S activations in the downstream part, and it will only lead to the activations of the CAMP, not the Ca2 plus. So whether one wants the this CAMP and Ca2 plus both activations or just the CAMP activations, it can be done through this synthetic PTH VHH ligand, or only the PTH ligands can be used for this CAMP and Ca2 plus activations. So this will give us more customization ability depending on the need of the experiment or treatment. So I hope you understood how these receptors activations can be affected by this induced proximity, and some of the examples are shown here for better understanding. If you need to understand more about each of these topic in detail, you can go through the reference of this review paper in the description. So that's all for this video. Thank you for your time.